Welcome to Lesson 13.3, Mutations. In this lesson, we'll define mutations and describe the different types of mutations. We'll describe the effects mutations can have on genes. Mutations are heritable changes in genetic information. The enlarged shape of the flower shown here is caused by a mutation that affects the growing regions of the flower tissue. Mutations come in many different forms. The flower shown on the previous slide and the lion shown here are just two of countless examples. All mutations fall in two basic categories: gene mutations and chromosomal mutations. Which type of mutation is likely to be more harmful? Chromosomal mutations is likely to be more harmful because many genes are found on one chromosome. Let's take a look at these types of mutations. Changes in a single nucleotide can affect the amino acid sequence of proteins. Point mutations include three types: substitutions, insertions, and deletions. You'll learn more about each mutation in the coming slides. Point mutations generally occur during replication. If a gene in one cell is altered, the alteration can be passed on to every cell that develops from the original one. So what can a mutation do to the outcome of transcription and translation? Mutations change the code. This changes the mRNA code. This changes the amino acids in the protein, and this changes the protein structure. This can also affect the protein function. In substitution mutations, one base is changed to a different base. Substitutions usually affect no more than a single amino acid, and sometimes they have no effect at all. Let's take a look at an example. Take a look at the second codon in this example. The codon is CGU, which codes for the amino acid arginine. An example of substitution: one of the C nucleotides is replaced with a T. This causes the codon to change to CAU, which codes for histidine. So the resulting amino acid sequence is changed, resulting in a change in the protein. Not all mutations result in a change to the amino acid. For example, if a mutation changed one codon of mRNA from CCC to CCA, the codon would still specify the amino acid proline. So not all mutations are harmful. Insertions and deletions can be much more harmful. Why do you think that an insertion or deletion could have a dramatic effect? Because the genetic code is read three bases at a time, if a nucleotide is added or deleted, the entire reading frame is shifted. Consequently, every amino acid after the mutation will likely be altered. Insertions and deletions are also called frameshift mutations. These mutations can alter a protein so much that it is unable to perform its normal functions. Sometimes a mutation affects an entire chromosome. Chromosomal mutations can change the location of genes on chromosomes and can even change the number of copies of some genes. There are four types of chromosomal mutations. In deletion, a portion of the chromosome is removed. In duplication, an additional region of the chromosome occurs. In inversion, regions of the chromosome switch places. In translocation, regions of chromosomes may switch from one chromosome to another. So what is the difference between inversion and translocation? An inversion mutation reverses the direction or part of one chromosome. A translocation mutation attaches part of one chromosome to another chromosome. Mutations can be minimal, beneficial, or disastrous to an organism. Some mutations arise from mutagens, chemical or physical agents in the environment. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. Physical mutagens include some forms of electromagnetic radiation, such as X-rays and ultraviolet light. If these agents interact with DNA, they can produce mutations at high rates. Cells can sometimes repair the damage, but when they cannot, the DNA base sequence changes permanently. What would be the effect of a compound that interferes with base pairing? It would increase the error rate of DNA replication. Other compounds can actually weaken the DNA strand, causing breaks and inversions that produce chromosomal mutations. Some of the most harmful mutations are those that dramatically change protein structure or gene activity. The defective proteins produced by these mutations can disrupt normal biological activities and result in genetic disorders. Some cancers are the product of mutations that cause the uncontrolled growth of cells. Sickle cell disease is a disorder associated with changes in the shape of red blood cells, as shown in this image. Sickle cells are crescent and star-shaped. Some of the variation produced by mutations can be highly advantageous to an organism or species. Mutations have helped many insects become resistant to chemical pesticides and have enabled microorganisms to adapt to new chemicals in the environment. Plant and animal breeders often make use of good mutations. For example, when a complete set of chromosomes fails to separate during meiosis, the gametes that result may produce triploid or tetraploid organisms. The condition in which an organism has extra sets of chromosomes is called polyploidy. Based on this information, why might plant breeders want to encourage these good mutations? Well, changes to the ploidy numbers of citrus plants can affect the size and strength of the trees as well as the quality and seediness of their fruit. Polyploid plants are often larger and stronger than diploid plants. Important crop plants, including bananas and limes, have been produced in this way. Take a moment to create a table in your notes that identifies the major types of mutations.